Welcome everyone to the June 2025 edition of the ARC seminar. We have two exciting speakers today, Rohit Singh from Duke University and Joshua McGrath from Weill Cornell Medicine in New York. To briefly introduce Rohit, he's an assistant professor in the departments of biostatistics and bioinformatics and cell biology at Duke University. His research interests are broadly in computational immunology, ranging from regulatory inference from single-cell multiomics to the design of new proteins and small molecules for drug discovery. And his recent focus area has been the investigation of biological foundation models to interpret and leverage their learned representations, which is a really important problem in the field. And today he's going to talk about the application of foundation models to B-cell receptor repertoires. Looking forward to your talk.
Thank you, Victor. Thank you, folks, for having me. Really excited to be here. So in this presentation, I'll be talking about analyzing immune repertoires and sort of some of the issues that you run into and how can you address those issues scalably and get the best insights. So to start off, I mean, this is a figure taken directly from the Antibody Society, and this is a reflection of the explosion in the field of immune repertoire sequencing over the last few years. Started off with single cell sequencing, and very quickly we realized we could do immune repertoires. And there has been, I would say, almost an exponential growth in the amount of data we are collecting here. And if the trends hold, and if we are able to get to, like, say, beyond the $100 genome to maybe even cheaper, you could imagine maybe in 10, 20 years, each of us during a regular physical might have some blood drawn and some circulating repertoire, at least noted for later assessment and benchmarking against when you get infected or when you are sick. So there are lots of things you can do because you're essentially getting a view into your immune system. I mean, some of the things that I'm most excited about, I'll just give you a few examples. So here, for example, I'm showing you 3 cohorts, hence the slightly different colors of individuals. And let's say there are some folks who have been infected by different strains of a virus. And some of them are protected against one strain, others are protected against another strain, and so on, because of their innate immune, not the innate, their adaptive immune system. But also, let's say there are super responders who happen to have an immune system that gives them protection broadly against different viral strains. So in principle, if you could identify these super responders, you could then look at the overlap between their immune coverage and how it distinguishes from other people's immune coverage and hone in on broadly neutralizing antibodies. And I would argue that this approach of trying to find broadly neutralizing antibodies is probably more effective than taking a purely computational de novo design approach to antibody, because here you are getting a humanized antibody that is actually neutralizing. It's not just about binding, and that is broadly protective. The challenge, of course, is that these are different individuals, different cohorts, different backgrounds, all of that. Other places where having a better understanding would be helpful is, for example, if you want to understand cross-protection or cross-reactivity, where you have 2 individuals, one who had prior exposure to a different antigen, and they're both exposed to the same antigen. And the one with the prior exposure doesn't get sick or is protected. Why? Because this helps us understand what kind of crossover can happen. More broadly, babies and old folk have weaker immune systems. Why? Is it because they have fewer diversity, or is there like new classes come in? What's going on? If you can understand that, we can help address immune decline in older folk. All of this is things you could do if you could better understand and work with immune repertoires, right? More recently, there was this really cool paper from the Boyd and Kundra labs which essentially said, let's take a bunch of B cell receptor sequencing repertoires, T cell receptor sequencing repertoires. They built three kinds of models, one based just on sort of clonal characteristics, one based on CDR3 sequences, and one based on sort of language models fitted on individual pieces of the V genes, or fitted for each V gene. And what they were showing was surprisingly strong performance in being able to predict the disease status of an individual by just the repertoire. And so essentially the insight is that there are signatures here which could tell you. I mean, it's a black box signature right now, but there are signatures here which you could get, right? All of is the promise. What is a challenge? And this is one of my uh, other papers, favorite papers in systems immunology. This is Briney et al. And here they looked at about 10 individuals and sequenced them, and they looked at the clonal overlap. And the striking thing for me in this figure was that if you look at the clonal overlap that is seen across all 10 individuals, that fraction or that percentage is actually 0.02. A very tiny fraction of clonotypes are seen conserved across all individuals. And if you think about it, that doesn't make sense. That doesn't make sense because almost all of these individuals have shared stressors. They almost all have had the cold. They probably have had similar vaccinations. Many of them must have had the flu. And yet, when you look at their immune signature, you basically are not seeing that. And that comes to sort of the running theme of this presentation, is that the issue is not so much that you're not seeing overlap, is that you're not measuring it correctly. You don't have the right representation to capture the functional overlap that you have across these individuals, even if you don't see a sequence overlap between them. And going back to the Zaslavsky paper that I talked about, and this sort of stands out there as well. So they find very strong performance in terms of predictive accuracy using different models. And in particular, a model that basically uses this sort of clonal information with some overlay of somatic hypermutation rates does really well. So this is both good and bad. It's good because we know that certain V genes confer protection a bit more on certain infections. So you expect that. But at the same time, I just showed you a figure where effectively, if you look at the sequence scale, clonal overlap across individuals is basically close to zero. So what this means is if I give you a new individual who has a different clonotype signature, you basically will not be able to say anything about them. Right? So and a semblance of this shows up in the paper itself as well, where they point out that if you were to use only age, sex, and ancestry, kind of like proxies for different clonal signatures, that thing itself is a remarkably good classifier. It is not as good as the repertoire models, but it is a good classifier on its own. So in some sense, the way I interpret this paper is that it's giving you an existence proof that immune repertoires carry enough content for you to tell out like which diseases the individual might have been exposed to. But it's not an existence proof like mathematicians would say. You can't easily sort of tease out this is what is driving it. And again, this goes back to my point about having more effective representations. So the last thing is more of a technical data processing challenge, is that the way many of these sequences are assayed, you have primers associated typically on the N-terminal side of the protein, but it's a DNA sequence, but yeah, or the C-terminal side. And the bottom line is that most sequences you get are slightly truncated. And to clean this up requires a BLAST or IG BLAST-based pipeline that scales poorly as you sort of go to hundreds of thousands or even millions of sequences. And again, if we had better, more effective representations, we could maybe do this more efficiently. So that's the setting. And so this is where we are interested in. And just by a very high-level overview of what my lab is interested in, as Victor mentioned, um, we're interested in questions at two scales. One is at the molecular scale, and there many of the questions we look at basically boil down to the insight that the growth in sequencing data has far outpaced the growth in, say, structure or function data, even though the latter is what we often care about. And can we come up with methods which can leverage all of this sequencing data, often poorly annotated, to be more effective at answering questions about structure and function or even design? A second set of questions my lab looks at is more at the system scale, where we look at, for example, single cell omics, multi-omics, spatial omics, and essentially say, can I recover dynamics and regulation and communication from these things? And one reason why I really like the project I'm sharing with you is that it spans both these scales. And that's rare in biology, where you're able to work multi-scale in a single project. And the idea here is that the the core atom of every repertoire is this individual receptor sequence, which is at the molecular scale representation. But what we are trying to do is take these molecular scale representations and aggregate them in a way where you can start to answer systems level questions with this. And the approach we use, okay, actually, before I tell you the approach we use, let me sort of give you the one slide punchline as to how we go about doing this. And so I'll be showing you a representation scheme where essentially we take the same sequences that I showed you in the Briny data, except that now in this new representation scheme, if you plot all of those entities on a two-dimensional PCA, and the colors indicate density here, what you find is a remarkable overlap across individuals. And the claim here is that this new representation is capturing some aspects of structural and functional overlap across individuals. And we're exposing that there is a shared structural and functional coverage across these individuals. And I'll try to support that claim with some evidence as we go along. So the core thing that we work on, that we use here, are protein language models. So these are models that rely on the same technology that ChatGPT uses, and in very similar intuitions, actually. So part of the insight here is that we have a ton of sequencing data. So on the left here, you are seeing the growth of sequencing data in RefSeq. The y-axis is on log scale. So what you're seeing is essentially exponential growth that has gone on for 20 odd years. And so you now have hundreds of millions of sequences. And you can use this to essentially train models that do not need any additional annotation. They can build their own training labels from the data itself. And they come up with really powerful representations that you can use out of the box, or you can adapt them for a specific task. Now, one distinction between ChatGPT and, say, protein language models is that in ChatGPT, the interesting task is usually next token generation, which is I give you a sentence, tell me what the next thing should be. In biological realm, especially in proteins, I think the more useful context is typically thinking of it as representations, because you're not so interested in adding one more amino acid at the end as understanding the overall structure and semantics of this entity. So just many of you probably know what protein language models are and how they work, but I'll just give you maybe a 3-minute overview on this. And the way protein language models work is a bit like how we teach kindergartners to read. So there are two philosophies to teaching kids to read. One is when you read, you should, so for example, for me, English is a second language, right? One philosophy is you read a book, you sit down with a dictionary. So when you see a word you don't understand, you look it up. Nobody does that. The approach that usually works better is just read a lot, and every time you see a word you don't understand, you try to guess its meaning. And the more you read, the better you'll get at understanding those words' meaning. And that is what masked language modeling is. So here, for instance, if I hide the word, I went for a dash around the block. Um, Unless you're in Venice, you're not going for a swim around the block. In fact, even if you are in Venice, you should not go for a swim around the block. Um, you're maybe going for a walk or a run around the block. And the same intuition can apply in protein sequences. I can hide a few amino acids and I can ask you to guess which amino acids would fit in here. And you would essentially come up with statistical rules to saying, okay, this is what works. So far, so good, right? Like, I mean, none of this is something that you could not do 15 years ago. What has changed is new machine learning technologies like the transformer that let you incorporate long range interactions. So, for example, if I change the word block to track, then you would probably agree with me that run becomes more likely, so that you are more likely to walk around a block, but run around a track. And this kind of long range interaction is what became possible with the transformer technology and all the advances that has happened. And this is relevant for proteins as well, because this essentially is picking up on coevolution, where two entities, two parts which might be coming together in structure, are far away in sequence, but together in structure, you kind of understand their interaction. And what you get from an analysis perspective is you train all of these models on these millions and tens of millions of sequences. At the end of it, for any sequence, you can feed it into the pre-trained model, and you will get back a representation. And it will be for sequence of length n, it will be a n by 1000, 1000, depending upon the model matrix. And the key thing is that that representation captures both local and global context. So for example, the two leucines here, they are going to be having different representations because they're capturing the context of each of these leucines independently. So sort of another way to think about what uh, PLMs can do for you is to essentially think of it as a comparison to multiple sequence alignments. So PLMs are essentially trying to learn the rules of evolution, right? Like evolutionary conservation. And that is what a multiple sequence alignment also does. But the thing about a multiple sequence alignment is you do it one protein family at a time. You have to find similar sequences, you have to line them up, and then you look at conservation per column. Protein language models are like the movie Everything Everywhere All at Once, right? Like, so you take all proteins, you don't even care about families, and you look at all possible combinations across columns, right? So you could have, you can make MSAs more sophisticated by looking at pairs of columns, and that is called like the correlated mutation analysis. But that soon starts to get combinatorially complex. PLMs, by essentially sort of folding that long range interaction into the transformer architecture, are able to look at both short range and long range interactions. So the flip side is that you don't know what they're picking up. So you have to do a decent bit of work and put some careful thought into extracting the features that are relevant to what you care about. And a great part of what I'll be talking about is exactly that. One more thing I want to sort of point out is that it's an happy accident that somehow the statistical mechanisms that the PLMs learn seem to mimic the evolutionary mechanisms. So for example, uh, if you look at this sentence change, where I change the word chicken to fish, a natural language model will basically score the second sentence as being not a very likely sentence, not a fit sentence. You could do exactly the same thing with protein sequences. You can make a mutation and you can say, and the language model will say, the protein language model will say, well, this is not a very likely sequence. It's not a fit sequence. And turns out this statistical concept of fitness actually recapitulates the evolutionary concept of fitness. So that if you look at human genetic variant data using language models that have never seen human genetic variants, they will still do a good job of scoring pathogenic mutations as being the ones that are statistically unfit. That is really cool, but yeah. So with that, now let's talk about how you would apply these to antibody repertoires. And here you have two choices. You have an approach where you can essentially take off-the-shelf foundational PLMs trained on basically general purpose proteins and use them. Or there are methods where folks have basically taken just antibody sequences and essentially applied the same technology and trained the language model. So the advantage of the latter is that presumably you can be more data efficient because essentially you're just learning the antibody specific features. But that is also a downside because the foundational PLM is learning these general purpose features, so which your antibody specific model might not be seeing. On the other hand, the foundational model has the weakness that it relies on evolutionary conservation, and there are regions in antibodies that are explicitly not conserved evolutionarily. So is there a middle way? And that is what we did, which is we designed a new language model, actually really a layer on top of any language model that lets you sort of have the best of both worlds. So the setup is that you can adapt any existing foundational model for BCR sequences. By the way, all of I'm going to be talking about is for BCR sequences, but the core idea is applied to T-cell receptor sequences as well. And we build models separately for heavy and light chains. In many of the results I'll be showing you results for heavy chains, but like sort of corresponding things work for light chains. And the key idea in AbMAP is that you can, just given the sequence of a BCR, you can predict where the CDRs are likely to be. And you can do this using some methods from Charlotte Dean's lab, NRKey, and this new, which I'll call NRKey2. And because we want to leave some leeway, we essentially will include 2 framework residues on each side. So you can focus on just these parts, and we try to make our representation be really good at on these parts. So one way we do that is by essentially sort of doing a counterfactual, saying, what if these parts had some other sequences? And can I subtract out that background from this? So I'm focusing on what specifically is in these specific CDRs compared to some other CDRs. And the next thing we do is essentially then superize it with structure information and binding specificity information. So we use antibody structures from PDB, and we say our representation should mimic structural similarity. And we use in the paper setting binding data from Libra-seq, and we said, okay, the representation should optimize binding preferences for BCR sequences. So the overall setting is essentially, if you do this, then you can have a representation that you can use for a variety of different things. You can even use it for structure prediction, though I think other methods for structure prediction might give you more flexibility. And in particular, we find it especially useful for analyzing repertoires. By the way, this work was jointly with Chiho Im, who was then an undergrad at MIT and is now a grad student at Stanford. And so for those of you interested in the technical details, sort of the way it works is we start off with a sequence and we take a pre-trained model. It can be any model. And you get the representation for the entire full length sequence. Then we will use NRKey or NRKey2 to sort of identify the locations that are the CDRs plus 2. And we'll come up with just hypothetical counterfactuals for sequence changes there. We'll subtract those out. And then just subset out the CDRs. So that gives you a variable length representation. Then we pass it through transformer layers that are going to do the structure function supervision. And the structure function supervision happens through what's called a Siamese network. So a Siamese network essentially you have the same model, two copies of it. So the parameters are shared. And you essentially pass in two sequences and you say something about how similar you want those sequences to be. So we have two tasks here. In one task, we take pairs of PDB antibody structures, and the supervision is on the TM score of the alignment of those two. You can do it on the whole antibodies, the heavy chain thing, or you can do it on just the CDR3, gives similar results. And the second supervision is on Libra-seq similarities in binding profiles. And you essentially take your concatenated thing and you essentially are piping it through a couple of layers of transforms, not very deep, and then just trying to optimize this. So this works well. Before I tell you about how well it works, I think there's one thing I want to share, which hopefully will be of use to all of you in your own work. So a particular pre-processing challenge we ran into is that as we get these truncated sequences and we wanted to fill them out, and it's crucial that you fill these out for language model representations because a language model representation of a truncated sequence can be very different from the language model representation of a non-truncated sequence. And so sort of the standard approach would be to use BLAST or IgBLAST, estimate sort of like the clonotype effectively, and then impute what the missing N or C residues would be. You can actually use a language model to do this much faster. So the key idea is that NRKey, the CDR numbering program, is good enough that it can actually mark out missing locations on the N and the C termini. And then you can use a foundation model. So in this case, we used ESMC, which is sort of one of the more recent ESM models from the ESM folks. It's not a very big model. It's about 600 million parameters, so it fits on most GPUs, and it's free to use. It doesn't have some of the restrictions that they have on the ESM3 family. But really, you could use any ESM here, I think, or any language model, and it will work reasonably well. And this gives you a 10x speedup. This work is led by Jiazhang Mao, who is an incoming grad student in my lab and who has been working with us before this. Some of these things I'm showing you are unpublished. I know this talk is going to be on YouTube, but still just letting you know. So here, what I'm showing you is that Anarchy's numbering is effective at identifying that these locations are missing. And as you would expect, because of how the primer tagging works in these sequencings, you often miss a good chunk of the N terminus. So you want to get that, and sometimes you'll miss a bit of the C terminus. And then you can use ESMC to go and impute these locations. And it does a pretty good job, like an 85 to 90% accuracy in recovering missing residues. And remember that even if you were to do BLAST or IgBLAST, they will also have some failure rate because some of those cases, they will not be able to give you a high quality prediction of this is the one thing you should be replacing with. And by using this approach, what we're able to do is essentially take our truncated incomplete sequences and make them near complete. So effectively, what you're seeing here is a heat map where orange is before and blue is after. And you're seeing that, especially, for example, the proportion of entities that have full V-gene coverage, which would be on the N-terminal side, is low. And by imputation, we are able to make it pretty close to 100%. So that's an effective thing, I think, that you could use in your work as well. Okay, going back to AbMAP, right? So there are a few standard things that these language models are being used for. And one of them is broadly what you would call property prediction. Basically, you give me a few examples of some label or some property, and then I give you, for different mutations, what that label or that property would be. In this case, the properties are predicted energy values for variants of M396. And what I'm showing you in this graph, it's in the paper as well, is essentially that using AbMAP on top of a foundation model gives you substantial power. And this power is especially meaningful when you have very limited data. So essentially, if you're looking at regimes where your training data is 1/100th of the test data, the relative outperformance over simply using, for example, one hot amino acid encodings gets strong. Another thing that we used AbMAP for, and this is another common use of language models for antibodies, is to essentially uh, direct an optimization campaign. So in this case, we started with this data from Engelhardt et al., where they ran a campaign to identify SARS-CoV-2 peptide binders. And they published a library of 4 libraries, each where they essentially had the wild type, and then they explored all the 1-point mutations and some of the 2 and a very few of the 3-point mutations that would be possible. And our goal was, can we use, and for each of those, they had essentially some experimentally measured binding score. And our goal was, can we use this to design 3 or 4-point mutations that would be good? And again, the basic approach here is you would essentially propose a mutation, and then you will score it using some kind of an AbMAP model. And then you would say, if it's good, I'll use it. So one thing here that I want to draw your attention to is something that you probably have seen if you have ever done this kind of an optimization campaign, is that when you're coming up with these candidates, you're trying to balance a few different objectives. You want to find candidates that are going to be high performant. But you also don't want to put all your eggs in one basket. You don't want to, even if your model says this mutation is the best, you don't want just that mutation all the time. And you want something that will work reasonably well, even if it has small changes. And so people have tried doing this with some kind of Bayesian optimization or other techniques. I'm presenting to you something that is, I think more robust and works better on a variety of cases as we have tried. And the first thing in that is that instead of building one scoring function, you build an ensemble of scoring functions. So for example, you can build a regression scoring function, which is built based on delta delta G, or you can do cutoffs on your KD, and you can have classifiers. So you build an ensemble of models, and then you generate a whole bunch of candidates. And this is the power of these language models, that you can basically generate 1 million candidates and work through this. It's just compute time. And you score all of these candidates using all of your ensemble models. And then you essentially sort of take the intersection of top hits across these ensembles. And you sort of calibrate your intersection so that you end up with maybe 10 or 20x more than what you will eventually test. You take that large set, and you cluster it. So we did k-medoids clustering. You can also do k-means clustering. And the number of clusters you choose is the number of assays you want to perform. And then from each cluster, you sort of take the central point and use that as the candidate sequence. What this does is that it gives you all the 3 things you want. You're picking high scoring hits. You're picking diverse hits because you're picking individual clusters, and you're picking something that is robust because instead of picking something at the boundary, you're picking a central entity in the cluster. And so this is just an indicator from one of the libraries where the left-hand side is sort of the scoring function. The right-hand side are the cluster colors that from our top hits. In black star is the wild type. And then essentially from each of these colors, we pick sort of the central point and we score that. So this general approach we have found works broadly well in other settings as well. So once with this, then we worked with Sanofi to essentially who sponsored this work to validate this. And it works pretty well. And I'll let you read the details in the paper. Because, okay, so I want to get to this part, which I think is a distinguishing aspect of AbMAP and a general approach to sort of analyzing repertoires. So one way I want to motivate what I'm going to show you next is essentially pose this problem of disentangling study-specific effects with disease effects. So if you're familiar with single cell literature, there is this tension that people have in terms of saying, okay, I want to regress out batch effects or something. And so batch effects here in an immune repertoire sense are not so much about data quality because you don't really have dropouts, especially once you've done the imputation, but more that each study has a bias in terms of the individuals it recruits. And those individuals have age, ancestry, gender biases, which basically make these studies distinct. And so the way we address this is we essentially curated from the observed antibody space dataset 4 studies, each which has both healthy and COVID individuals, good patients. We took 3 of each, and then we subsampled their repertoire so that they're roughly equal sized. In some cases, the repertoires were slightly small, so we couldn't subsample it. But the overall point is this gives you an apples to apples comparison. And now we're going to look at a variety of different representation schemes to see if we can use the representation scheme to predict healthy versus COVID, or just the study, one of 4 studies. And the goal is that you want the representation scheme that is really good at predicting the condition and really bad at predicting the study. And so let me show you sort of a baseline. So this is ESMC, which is basically a state-of-the-art model. And what I'm showing you here, every dot is an individual. So it's a collection of BCRs in each dot here. And the plotting here is done using an optimal transport-based distance between individuals. I can talk about that later. But the takeaway, very stark takeaway from this is that the model is actually better at distinguishing studies than it is at distinguishing condition. So on the left, I'm plotting those points by their study. On the right, I'm plotting it by their condition. And this is sort of what I was getting at when I said a model that makes predictions using clonotypes. It's good, but it's also bad, because if I give you a new study, which would have different clonotypes, how do you work with that? And so the question is, why are study-specific effects so prominent in these PLMs like ESMC? And by the way, I would claim that this would be true also for antibody-specific PLMs, because in fact, I would argue they have a larger risk. We haven't measured this yet, but I would argue they have a larger risk of this because they are focused only on antibody sequences. So they might be picking up the clonotypes even more. So how could you address this? One way you could try to address this is essentially, again, sort of inspired by single cell literature in terms of sort of adjusting for batch effects. So in this case, what we do is I have the sequence and I can run this through my regular PLM and get an embedding. Embedding is the representation. But separately, I can essentially get the VDJ markers for this, like the clonotypes. And I can take just that as the input and train a small neural network that tries to recover exactly this embedding, right? And if I do that, and the whole point is that you can actually do this with some accuracy and surprising accuracy. And then if I can do that, I can subtract this second guy from the first. And presumably what I will have is something that has less of the clonal signatures in the representations. And so here I want to show you why something like AbMAP is useful. So the setting I'm showing you is essentially I have the foundation model embedding, and I have taken a variable length embedding and I have average pooled it. And I also have a tokenization of its clonotype, essentially like a numeric representation of it. And I'm trying to predict the clonotype from this. What I'm showing you here is that you can get a very high R square in predicting the clonal signature of a BCR just from the ESMC thing. And which kind of makes sense. But the second thing that should surprise you a little bit is that if I take this entire thing and then I essentially extract just the orange bits and average over only the orange bits, you would naively think that that should take out the clonal signature. It does not. It actually remembers the clonal signature about as well as the original thing. And part of the reason is that PLMs are extremely redundant representations. So simply subsetting on CDRs is not enough. And this is sort of a second theme that has been going on through PLM literature over the last year or year and a half or so, is that PLM representations, both in the dimensional axis as well as along the length axis, are highly redundant. So in particular, like a regular protein language model representation would be length by, say, 2000. We and many others have shown that you can just squish everything across the length, get a fixed length thing, and it remembers a lot of properties about the protein. And in fact, if you, rather than squishing it to one column, if you squish it to 50 columns and do some more work, it actually remembers enough that you can regenerate from this. And so this is work that postdoc Kapil Devkota in my lab did on protein redesign, where you can add insertions and deletions and mutations. Separately, you can keep the same length, but you can squish the dimensionality, and you can still do that quite effectively. And this was shown by Amy Liu and others here. So the key point is, if you if you predict the entire sequence and you just take a subset, that subset will still remember a lot about what's going on. So you have to do a bit more. And a bit more here is that you essentially, so one of them is AbMAP, but the other one is a cheap thing that you can do with ESMC, which is you essentially replace these orange bits, which are the CDRs, with gaps. And you essentially give this partial sequence to ESMC, and you subtract out this partial representation from this. And now you notice that the ability to predict has gone away to a large extent. And AbMAP sort of takes away that ability even. And by the way, one issue though is that even though you can regress out the clonotype linearly, it seems that the study specific effects don't go away entirely. So this is ESMC, where the original ESMC, where we regress out the clonotype. You'll notice that these two studies, which were distinct in the previous figure, are now a little bit merged, but on the whole, the study separation is still very stark. And this is something that AbMAP addresses. And so in this figure, I am essentially showing you the trade-off of study accuracy versus disease accuracy. So along the x-axis is how accurate each representation is at separating studies. And here you want to be as low as possible. Along the y-axis is how accurate the model is at predicting disease and here condition. And here you want to be as high as possible. So essentially this star is where you want to be, which is maximally accurate on the disease and basically random on the study. What you actually see is that a regular protein language model and even the CDR subsetting of it is good at distinguishing diseases, but it is actually very good at distinguishing studies as well. And if you just do the contrastive thing where you just subtract out this one part, that reduces the study separation, but it gives you a pretty big hit on the disease. And AbMAP sacrifices just a little bit of disease separation accuracy, but saves a lot on the study. So it reduces the study effect a lot while broadly preserving the disease accuracy. And this you see in the actual representations as well. So this is AbMAP before regressing out the clonotype signal. So you see that the studies are pretty well distributed here. It's not easy to sort of separate them out. If you regress out the clonotype signal, and if you remember, the R square wasn't good at all, so there is not much that happens. But yeah, you can sort of see that the disease signal actually is slightly clearer to see than the study signal in AbMAP. And the key thing here is that one of the things that helps a lot is even compared to the ESMC contrastive step, where I just subtracted out the part that does not have CDRs, AbMAP does better. And the key difference in AbMAP versus that ESMC thing is that AbMAP was further supervised on structure and function. And this is a figure from the paper where we basically show that as you start from the raw embedding, the subsetting, and then the contrastive, the last step where you supervise is critical in giving you sort of the ability to separate these sequences in their ability to capture structure and function. And this now takes me back to the punchline I was showing you earlier, which is we took the briny data set and we essentially represented them with AbMAP, plotted everything in 2D space with PCA, and then colored it by density. And what you see visually is a very striking overlap, that there is a hotspot and then everything is sort of around that, right? And you can make this a quantitative statement as well. So essentially what we did was we said, in this case, we had 9 individuals we were looking at. We said, let me take a random BCR from one individual. And under your representation scheme, let me find the closest BCR across all 9 individuals. What is the likelihood that the closest BCR came from the same individual, right? And if your individuals are well mixed, then this likelihood should be 1/9, because essentially the closest BCR could have come from any individual. But on the other hand, if you have very individual specific representations, then the likelihood that your closest BCR came from the same individual is higher. And that is exactly what you see here. You see that AbMAP-based representations essentially are places where the closest BCR could have come from any of the other individuals. While if you use sort of a sequence similarity-based thing, then the closest individual is typically you yourself. The other cool thing, which is relevant translationally, is that if you look at antibodies that are used therapeutically and you plot them on the same space, they all end up landing in the hotspot that we are discovering here. And that is really cool because this gives you sort of the Lipinski's rule of 5 version for antibodies, where you could go and say, okay, how do I make sure that this is an antibody that will be well tolerated? Okay, so I'm going to sort of stop here, but I want to just sort of talk about what would be the future directions for this kind of work. So I think the challenges in analyzing B-cell receptor repertoires are pretty substantial. But if you can do that, it'll be a huge win for the field in terms of being able to get insights. The first thing that I spent some time talking about today is essentially how do you go beyond the clonotype signals that can confound what you are seeing. So the point here is not that you should not use clonotype signals. The point here is that you should disentangle them. So you can just have the clonotype signal by itself, and you can have the non-clonotype signal by itself. And you can still use the clonotype signal, for sure. It's not like you should not use it. However, what I also pointed out is that even after taking out the clonotype signal, there are study-specific things that you see in, for example, ESMC. And I think there are always going to be these cases where if there is, for example, a new epidemic and you sample a new cohort of individuals and you want to understand what's going on, in the moment, you're not going to have a genetic history of those folks if you're not going to have a vaccination history of those folks. And if you want to understand what is it that is distinguishing their immune coverage from whatever else you might have as a reference, you need to have a way of being able to figure out the confounders and remove them. The challenge is you don't even know what the confounders are. And lastly, as you sort of think about figuring out what are the disease signatures in a immune repertoire, there are sort of two models that could be plausible, and maybe both are true, which is one is that there is some kind of sort of the analog to clonal expansion, that certain repertoire clusters become more dominant. Or the other is that completely new responders show up. Now, because of the sampling issues, it can be hard to distinguish these two, but having the ability to distinguish these two is also going to be important. Okay, I'm going to stop here. So a lot of this work more recently was with Jiazheng Mao. I talked about. I want to thank a whole bunch of folks, our funding agencies, and we are recruiting. We are recruiting grad students as well as postdocs. So please reach out. And happy to take more questions now.
Thanks so much for this very interesting talk. I have questions. And we only have 8 minutes, so we should just keep questions and answers rather short. Um, for the imputation part, how
Mhm.
sure are you of the imputation quality, given that BCRs could be mutated to a certain degree? So how sure are you that mutations are inferred, or are you just talking about, I guess, more germline based?
More germline based, basically, for sure.
Okay.
So essentially, but that would be what we would have done with the blast anyways, right? So essentially, the baseline is if I do the imputation using IG blast, am I
Yeah.
matching it or not?
Yeah. Yeah. So you're not claiming that you can impute heavily mutated.
Yes, and the hope is that the N and the C terminus will not be seeing that heavily mutation stuff. That would be in the middle because
Yes,
the imputation is primarily on the N and the C terminus.
I see. Okay. Got it. Got it. And then I think you're really hitting the nail on the head with the batch effects, that this really keeps the field back. Um, from the studies that you looked at, I think there are um, several confounders, right? Because you have different patients and
Hmm.
different probably library preparation protocols probably also. So there are
Yep.
many different things that are varying there. What could be nice for this, I think, is this one TCR benchmarking paper but it's TCR, which was published in
Mhm.
Nature Biotech, I think, in 2021, where they had the same sample sent out to different labs with different library preparation protocols. So that could maybe be so. Um, what my question is, it wasn't super clear to me. What do you think are the batch effects? And what is
Yes.
dominating your signal? And you talked about clonotype, but of course they differ across different repertoires. So yeah. So
Yes. So I'm almost
yeah.
more worried about the latter, where even if it was the same sequencing technology, I
Yeah.
just had different studies, I would have different clonotypes.
Okay. So just the background
The
repertoire.
background, I think, is a so I think the study specific batch effects are important as well, but I
Yes.
have a feeling that that can be addressed. So my view there, and it might be wrong, is
Because we know
that's
that library preparation protocols can heavily influence which germline genes get
genes get
higher or
picked.
lower. So yeah,
Yes, that's fair.
yeah, yeah.
Yeah, I think, and that definitely could matter, but
Mm,
in some sense, the whole point is yes, that you get this biased representation, over representation of certain sequence patterns than others.
mm, mm.
And how do you sort of account for that in some way, or at least look beyond that?
Yeah, 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 yeah. But so where do you see this going? So do we need new experimental data where we have better control over these factors, or
I think
how do you?
yes, I think all of the above. So for sure, yes, that'll help. But I think the bigger thing here is one thing that immune sequencing has to some extent, which, for example, single cell batch effect field does not,
Mhm,
is scale.
mhm,
If I could give you eventually maybe 100,000 or maybe 1 million sequences per individual,
mhm,
can I then have algorithmic ways of sort of saying this is the distribution and this is the distribution, right? And presumably, I can look at some mathematical ways of saying that all healthy individuals should have some similar distribution,
But this we
something like
know
that.
from statistical models from the Walczak and Moral labs, right? With the generation probability. So you don't mean that.
I don't mean that. No, what I mean is essentially saying that I think more data will help, but
Mm.
where I'm coming from, better data will help is better algorithms will also help.
Yeah. Mm.
And so what I want to say is, let's say your uh, protocol has a systemic bias for a particular germline versus another germline.
Mm.
And now, if you grant me the assumption that both of these germlines are capable of generating a diverse set of structural or functional
Mm.
coverage,
Mm. Mm.
and the structural and functional coverage is what is important,
Mm.
then by looking at just one germline where you have which is captured, but I can look at the variations in structure and functional coverage across individuals and still be able to give you an answer as to whether this individual is likely to have risk of this new variant. And that's what I mean by saying that having more data points within each repertoire helps because I can expand that germline and algorithms which can take that germline and sort of say, okay, this is what I actually need.
I see. I see. Makes sense. I see. I see. Are there more questions for it? I don't see any. So thanks again, Rohit. Very interesting talk, and I think I will follow up in a separate email.
Thank you. This was great. Thank
Yeah.
you all for this.
So
Yeah,
much.
and if you have questions, please reach out. I mean, I'm new to the system semiology field, and I would love to talk more and collaborate.
Perfect. Thanks so much.